Hi, my name is Aaron. I'm one of the research scientists at the Francis Crick Institute. So at the moment, I currently work in the RNA virus replication lab. So we are looking at RNA viruses, of which is a massive famous one currently circulating in the world, including coronaviruses. So at the moment, my projects are based on coronaviruses. I wanted to give you like a quick sort of presentation of what I do on a day-to-day. So I wanted to kind of show you what I'm currently doing in the lab, and hopefully it's pretty interesting. So my first slide is looking at a standard virus. So this is the standard structure for most viruses. So they all would have some form of genetic material, whether that's DNA or RNA in the center. And this basically is their archive to create new viruses. And it also determines how infectious they are and how deadly they are and what they need to do when they actually infect the cell. So they all need this. The genetic material is usually protected by a protein shell, which we call a capsid, or sometimes a nucleocapsid. Now, this helps to protect the genetic material, but then also once the virus has entered the cell, it helps to position it and orientate it so the host or the cellular machinery can easily access that genetic material and help it to make proteins or to replicate itself. The protein shell is usually protected by a layer of fat, which we call the envelope. So this offers further protection. It also acts as a docking station for the other proteins, such as spike proteins and other envelope proteins. Now, you've probably heard quite a lot in the media that with viruses, they tend to be easier spread during winter seasons rather than warmer seasons. And that's because with the layer of fat, when you cool fat down, it kind of solidifies. So if you ever had animal fat and it gets cold, it gets very solid. So that when it solidifies, it helps to protect the virus. And during the warmer months, the fat is a bit more liquidy, so therefore it doesn't offer as much protection to the virus. So hence, you'll probably get more infections during winter rather than summer. The spike proteins and envelope proteins, these help the virus to infect its host. So these fool the cell receptors into thinking that it's a ally or a friendly thing to allow it entry into the cell. And once these proteins bind onto the receptors, the cell will bring it into it and then proceed with infection. So I'm currently looking at SARS-CoV-2, which is COVID, and I've got a picture here to sort of explain its life cycle. I'm not going to go too much into it because it's extremely confusing, but what I'm interested in is usually this section. So once the virus itself has actually gained entry into the cell, and it's put its genetic material into the cell itself and it's beginning to use the host machinery to start to replicate its DNA, replicate its genetic material, and start making its proteins. I'm focusing on this part. So I'm using a thing called antisensitive nucleotides, which is just an extremely fancy word for saying bits of DNA. So the theory is, if I can make pieces of DNA which can bind to the coronavirus DNA, I can then start controlling the coronavirus DNA. Now, with antisensitive nucleotides, you can do many things. You can, upon binding the target sequence, you can degrade the target sequence. So in theory, if I bound coronavirus RNA, I could degrade it. I could do also a steric block. So physically, binding the piece of DNA or piece of RNA would make it inaccessible for the host machinery to actually help replicate it or produce its proteins. So we can do that as well. And this basic picture is just showing what the different sort of things that you can do with antisensitive nucleotides. The other thing with antisensitive nucleotides is because they are synthetic, they're man-made, we can change the backbone of them. So rather than it being DNA itself, we can actually make them a bit more like a hybrid DNA. And if you change the backbones of them, they have different properties. So they can go to different parts of the cell, they can go to different parts of your body, they can go to different organs, they can have a big lifespan in your body, they can have a short lifespan in your body. So we can kind of manipulate the backbone so for those pieces of DNA to try and do many different things. So what my experiments are looking at is you basically have all of these compounds will be the pieces of DNA to the antisensitive nucleotides with different chemical backbones, and they are all targeting different parts of the coronavirus DNA. So what I will be doing is using something like this in the top right, 96 well black plate. I will then basically put some cells in all of these wells. So the cells I use are green African monkey cells. They'll grow for a bit. with the compounds, with the pieces of DNA in them, so they can hopefully absorb them in. And then on the penultimate day, I will then infect with virus. I will infect with COVID. So I would infect, it's besides the wells at the top, I would infect every well with virus. So hopefully what would happen is, if the compound is effective, or if the piece of DNA is effective against the virus, you would see less amount of virus in the well. If there's nothing stopping the virus, if the compound doesn't work, then the virus will have free reign to infect as much as it wants to. And at the bottom, I am using remdesivir as a control. So I know remdesivir does kill the virus. Therefore, if I add this to the wells and the cells at the same time with the virus in there, I know at a certain concentration that will destroy it. So you should be able to see it. So you end up getting something like this. So towards the left, the cells, they are stained blue. So you can see that the wells do have cells and the cells are alive, and there's a nice layer of them. There's hardly any gaps or anything. It's a nice layer of cells for the virus to infect. And when we infect with the virus, we then go in with an antibody, which has like a little pigment on there. And when the antibody can detect the virus, it will cling onto it. So that's why you can see the virus here in green. And if you look at the remdesivir controlled wells, like as I did before, at high concentrations, there is no virus whatsoever. But then once you start lowering the concentration, the virus starts to reappear again. And with my compounds, they all very much differ. None of them have totally wiped out the virus, but some of them have significantly reduced its ability to infect and replicate. So like this well here, this well here, this well here, compared to wells like the ones on the outside, which have nothing in there except for virus. When you compare those sort of wells, you can see how much difference my compounds are making. And when you start doing these sort of things with a microscope, you can start actually generating numbers and seeing how much percentage is this actually affected this replication by. And hopefully we can keep on fine tuning our compounds to get something which will hopefully end up totally wiping out the virus. And then once we do that, we can then start producing this to go into animals and then clinically trying it for humans as well. So I hope that's a little sort of my sort of week in a nutshell. I hope it wasn't too confusing. The last picture is like me and my old lab. So we do hang out sometimes as well. We had Christmas dinner together and everything. But this is my lab, so and so in the bottom right, this is the Francis Crick Institute. So hopefully when the whole pandemic is over, if you guys ever get an opportunity to come down and see the gallery and everything. I hope you have some questions. Thanks.